Good morning to all. Hello to everyone joining today's webinar. I'm Junaid Tantre, and as a co-convener of this event, I'm delighted to share such a diverse and engaged audience. Your participation greatly enriches our discussions and underscores our shared commitment to advancing in our respective fields. I look forward to productive conversations and collective learning as we embark on this educational journey together. SPSR is distinguished for its engaging webinars, further enhancing its status as a leading platform for continuous professional developments in the pharmaceutical sector. I'm confident that today's session will uphold its tradition of excellence, providing valuable insights from a respected speaker. So let's seize this chance to gain new knowledge, participate actively, and progress our field. Welcome once again, everyone. I look forward to a productive experience for all the participants who have joined us. We are gathered today to commemorate the 85th SPSR webinar on science technology transfer from lab to plant, a distinguished event hosted by Society of Pharmaceutical Sciences and Research under the esteemed guidance of Dr. Shashi Alok sir, National President SPSR. The society continues to play a pivotal role in fostering innovation and advancement in the pharmaceutical sciences. The SPSR is devoted to advancing the pharmaceutical field through a variety of strategies. Our mission is to organize conferences, the support family-related initiatives, and endorse programs tackling public health issues in India, focusing particularly on disease prevention. So we make it a priority to recognize and honor pharmacists whose notable contribution drive progress in our fields. At the heart of our mission is a commitment to spreading pharmaceutical knowledge, ensuring a steady and robust exchange of information about pharmaceutical knowledge, patient services. By striving towards these objectives, we aim to elevate the quality and impact of the pharmacy profession, fostering a proactive and collaborative community dedicated to excellence. It's a great privilege to introduce Dr. Gaurav Goyal, sir, Deputy General Manager, Transdermal F&D, Zydus Lifesciences Limited, Ahmedabad, Gujarat, India, as a distinguished speaker today. Dr. Gaurav Goyal sir's expertise greatly contributes to our event, providing substantial knowledge and insights. Sir's participation is certain to enrich our discussions and expand exploration of technology transfer. We are grateful for his engagement and anticipate on enlightening session. It's a privilege to introduce Mrs. Monika Sabharwal, ma'am, as the convener for today's webinar, who also serves as the founder and national secretary of SPSR and CEO SPSR Periodicals Private Limited. Her significant involvement and leadership in the pharmaceutical sciences and research communities are indicative of her commitment to the field. We are very grateful to have her guidance in today's session also. I am pleased to invite Mrs. Monika Sabharwal, ma'am, to warmly welcome our distinguished guest, Dr. Gaurav Goyal, sir. Over to you.
Thank you so much, Mr. Tantre. Hello, everyone. I am Monika Sabharwal, the convener of 85th SPSR webinar, and it is my absolute pleasure to welcome you all to today's session on Science of Technology Transform from Lab to the Plant. This webinar is proudly organized by Society of Pharmaceutical Sciences and Research SPSR as a part of our ongoing efforts to bring the high quality knowledge sharing platforms to the pharmaceutical and healthcare community. We are honored to have us today our esteemed speaker, Dr. Gaurav Goyal, sir, Deputy General Manager, Transdermal F&D, Zydus Lifesciences Limited, Ahmedabad, India. His vast experience and expertise in the technology transfer and the formulation development will provide invaluable insights into the transition of scientific discoveries from laboratory to the large scale production. I am delighted to share we have received an overwhelming response for this particular webinar with more than 2100 registrations from over 20 countries. This global participation is a testament to the growing importance of this topic and the trust that professionals and students worldwide place in the SPSR's knowledge sharing initiatives. This is the 85th webinar in the RSPSR series, a journey that has been incredibly rewarding. Over the time, SPSR has grown into a global platform for professionals, researchers, and students by continuously organizing high quality free webinars on various topics. We ensure that our content is aligned with the needs of industry and academia, helping the bridge the gap between the research, innovation, and practical applications. We are immensely grateful for the overwhelming response we received from the participants worldwide. The continuous feedback, engagement, and enthusiasm from audience drive us to work even harder to create relevant, insightful, and impactful sessions every time. SPSR remains committed to foster academic and industry collaborations through webinars, conferences, and MOUs with pharmaceutical institutions. We encourage institutions interested in collaborating with us to submit their proposals via our official website. Additionally, SPSR membership program has been rapidly growing, with many professionals and students from the pharmaceutical and medical fields joining our community. If you are interested in becoming a member, we welcome you to visit our website and be part of this knowledge driven journey. At SPSR, we always strive to bring a new, engaging, and industry relevant topics to our audience. We encourage participants to suggest topics and speakers that they would like to listen in future webinars. Our commitment to the excellence and our ability to bring together the renowned industry leaders, academicians, and researchers from across the world have been a key factors in the success of this series. Today's session will shed a light on the process of technology transfer, a crucial step in translating research from laboratory scale to the full scale manufacturing. This topic is highly relevant to today's pharmaceutical landscape, where innovation must be efficiently scaled up to meet the market and the regulatory demands. Before we commence, I would like to warmly welcome our National President, Dr. Shashi Alok, whose visionary leadership has been crucial in impressive transformation of the SPSR. I would like to extend my heartfelt thanks to the significant contributions made by Mr. Junaid Tantre, the co-convener of this webinar, and organizing committee members, Dr. Saurabh Kose, Dr. Pradeep Gulani, Dr. Sanjay Nagdev, Mr. Ankur Agarwal, and Mr. Rishabh Banod. Their tireless efforts have played a crucial role in transforming the vision of this event into a reality. Join the SPSR vibrant community of over 50,000 pharma professionals actively engaging across LinkedIn, YouTube, Facebook, and WhatsApp. Let's connect, learn, and grow with the top tier webinars and the exclusive content. Do not miss out this unparalleled opportunity to elevate your pharmacy practice. Stay updated by subscribing to SPSI YouTube channel and enabling the notification bell icon for instant alerts. Follow our journey in advancing the pharmaceutical sciences that will benefit the society overall. Once again, I extend my gratitude to the esteemed speaker, our audience, and the entire SPSR team for their tremendous efforts every time. Without further ado, let's dive into today's session. Please join me in welcoming the Dr. Gaurav Goyal sir, as he shares his insights on science of technology transfer from lab to the plant. Thank you, and over to you, Mr. Tantre.
Thank you so much, ma'am. I really appreciate for granting me opportunity to present this webinar. And now we will start a short introduction of our guest profile. So, with the profound profile, Dr. Gaurav Goyal sir is a distinguished pharmaceutical scientist with a 15 years of industrial and academic research experience in transdermal drug delivery and novel formulation development. Sir is currently serving as Deputy General Manager and Head of Transdermal Formulation and Development Department at Zydus Lifesciences. Throughout his career, Dr. Goyal sir has contributed significantly to pharmaceutical industry, having worked with the leading organizations such as Shilpa Medicare, Akums Drugs and Pharmaceuticals, and Rusan Pharma Limited. In addition to his industrial roles, he has also shared his expertise in academia, mentoring students at Institute of Chemical Technology, Mumbai, ISF College of Pharmacy, Moga, and MET Institute of Pharmacy, Mumbai. Dr. Goyal sir holds a doctorate and master's degree in pharmacy from Institute of Chemical Technology, Mumbai, and a bachelor degree in pharmacy from Jamia Hamdard, New Delhi. One of his landmark achievements include the development of India's first indigenously generic fentanyl transdermal patch, which was successfully commercialized in 2009. Over the years, Sir has led research and development in oral thin films, transdermal therapeutic systems, implants, nasal sprays, lyophilized injectables, sustained release topical, and oral solid formulations. A passionate advocate for pharmaceutical innovation, Dr. Goel Sir is a lifetime member of Indian Pharmaceutical Association, the Association of Pharmaceutical Teachers of India, and the Controlled Release Society Indian chapter. With his extensive experience and pioneering contributions to novel drug delivery systems, Dr. Goel Sir continues to drive cutting-edge research, technology advancement in the pharmaceutical industry. It's a great satisfaction and pride that we declare that we have succeeded 2,100 registrants globally, showcasing an impressive diversity of countries. Significant attendance is noted from the nations include India, Algeria, Angola, Bangladesh, Congo, Ethiopia, Iraq, Iran, Indonesia, Malaysia, Morocco, Nepal, Nigeria, Oman, Pakistan, Saudi Arabia, Syria, UAE, Uganda, and USA. We are deeply moved by the substantial and varied responses we have garnered. Our heartfelt gratitude is extended to each and every person who has taken a moment to participate in this significant event. Thank you for all joining us today. So let's make this webinar an insightful and enriching experience for everyone involved. We have our esteemed keynote speaker, Dr. Gaurav Goyal sir, with us. Welcome, sir. Your insights will be highly valuable, and I would appreciate if you could present them in your speech. Over to you, sir.
Good morning, audience, and thanks to SPSR to invite me on this worldwide international platform. The numbers are very much significant, more than 20 countries. This is very impactful number, and the magnificent number, more than 2,100 registrations, is a very big number, and I'm very much delightful and hopefully, I think it will be a knowledgeable session for everybody. I will also learn from you. And this is all throughout whatever I have learned during my 20 years of tenure uh, from m.pharm to today. I'm going to share with you uh, those all insights so that it will be helpful for you to do the technology transfer from your laboratory, from your college. I think the professors will be very much, it is the presentation is oriented with respect to professors as well as lecturers which are there, how to think, that is the ideology and the thinking ability to for the technology transfer to the industry, because first of all we should know how to do what is required by the industry, what data is required by the industry. That is all things we are going to cover in this particular workshop seminar. And I am saying it is a workshop because it's like I'm going to give you the practical, some case studies where we can going to learn how this particular project was transferred to the industry, what was their expectations from the my team, from me during the transfer. So those all things we are going to share. So I'm very thankful to Dr. Shashi Alok, who is the president of SPSR, and Mrs. Monika Sabharwal and Mr. Junaid Tantral, the co-convener of this. With this, I am going to start with my presentation. Thank you very much.
So before you start, I would like to request participants during the session. If anyone wants to put question, they can put it in the comment box in YouTube. So sir can answer at the end. Thank you, sir. Please, you can proceed.
Is it the slide? Is it
Yes, sir. It's visible. Absolutely visible, sir.
Can you
Please.
confirm? Is that the slides visible?
Yes, yes, of course, sir. It's totally visible. Please continue, sir.
Just a minute. Hello.
Yes, sir. It's visible. Totally visible.
Okay. Thank you. Thank you for the confirmation. Yeah. So today's session, I'm going to speak on the science of technology transfer from laboratory to the plant. I am going to be the pilot of today's flight. My name is Dr. Gaurav Goyal. I am presently working with Zydus Lifesciences Limited, Ahmedabad, Mumbai, where we are going to take the stations where we are going to take. It is what is the science of the transfer process, the method development and the transfer process of the methods. Few case studies we are going to see in this particular journey of flight, and the factors affecting the QTPP, the documentation part of the technology transfer, and how to do the technology transfer from laboratory to the plant. What is the minimum studies which is required? That is the art of transfer. That is we are going to see today. So the science from the very laboratory to the plant level, which is a very complex machines are there, very large scale machines are there, which from the very small scale machines. So what documents, how to implement that transfer process, that we are going to look at today. So with this, first of all, the very encouraging example, I want to start with this particular presentation of an oral tablet. We all know that crocin Calpol tablets, which is a flat circular tablets, whose weight is approximately 700 milligram, and they contain 500 milligram of API, paracetamol, acetaminophen, which is also known as. And more than 20 brands are available into the market, but still the challenge is difficult to swallow. So this challenges leads to innovations, and the micro labs did the very good innovation with respect to development of Dolo 650. If you see the shape, it is biconvex tablet, it is con oval shaped tablets. So what from the flat circular tablets to this, because the flat circular tablets usually difficult to swallow because they stick into the buccal mucosa because of its flatness. It gives the larger surface area for sticking into the mucosal layer of our mouth. So this oval biconvex tablets give the less area to stick to the mucosal, and this design support for the easy solubility to the patients. So this small changes, not even any change in the formulation, the weight is almost similar, but only with the change of the design, we can support with any idea. So that is, we need to understand what are the challenges to the patients, what are the challenges to the users, and with those challenges, we can certainly can manage, do some small twinkling towards the formulation, and then we can improve and we can capture the market. Now Dolo is almost like a world leader into the production of manufacture of paracetamol tablets. So they, the one more thing which they have did it from the 500 milligrams, they introduced 650 milligram uh, paracetamol tablets, because it was clinically proved that it is effective in almost all type of fevers, like the corona fever, the dengue fever. There are lot many fevers which are recently upcoming. So this 650 milligrams, the 500 milligram was not sufficient to produce very high level fever like 103 degrees Celsius, 104 degrees Celsius, whereas 650 milligram was clinically proved to control the very high level fever also, which is of unknown origin. So this is one of the very, only this from 500 milligram to 650 milligram, they did a very significant improvement in the dosage regimen. We see the science of technology transfer, which is more, almost it is very much known by all of us, that the research team involves into the research and development. After that, they do the invention. Invention is basically on the basis of, like just we have discussed, the it is basically to overcome the challenges which is there into the existing market. The evaluation was done by the evaluation team, that the whatever invention is done, what are the different critical process parameters, what are the CMAs of the excipients, CMA of the API, critical material attributes, what are the critical formulation variables, all those stability data of this particular invention. All those data is evaluated by I can say technology transfer. It is, there are two units. We all can understand that one unit is developing the formulation, developing the science, developing the innovation, and that is known as the sending unit, SU. And they are do the technology transfer from the sending unit to the receiver unit. So another party who is receiving the unit is the receiving unit, and these people are not technically sound people, but they have the money, they have the plant, they have the industry where you can do the technology transfer. You can prove that your technology can work on their machines and can be successfully marketed. So basically it is commercialization, it is by the receiving unit and you are the sending unit, right? So once it is evaluated, then there is a part which is known as IP protection, intellectual property protection, done by the, usually between the, in the collaboration between sending unit or receiving unit, or it can be by the sending unit. Then the marketing team, licensing team comes into the picture, where they also evaluate the technology, and then it is a scale up product development, scale up on the machines, and after scale up it is the registration by the registration approval by the regulatory agency, and then after the approval it is public use and economic growth. So this is the complete cycle of technology transfer, how it works. And the continuous improvement is always there, because certain difficulties is always there into the production that can be rectified. So it is a continuous improvement. The formulation is always there. So one of the very, very common technology transfer which we see generally in a daily life, it is people, ladies of the house, and the cooks generally refer the recipes from the YouTube, which is very easily accessible, and then they cook the food. Now most of the people have started from the Corona period, 2020, they started cooking into the home. They realized that they can do that, and it is a very simple type of technology transfer, which is a very common technology transfer from YouTube for the cooking process. But how this is very much different from the pharmaceutical technology transfer, that is we are going to study. But these are the examples where also we can see that we are not getting the consistent quality and taste every time by the same recipe. If you cook today one dish from the same recipe, and after 10 days you are cooking the same dish from the same recipe, you are not going to get the taste. So we can understand what can be the problem with the inconsistent taste, because it is a variability due to the inconsistent raw materials. The vegetables which we used before and now and afterwards, it will not be consistent quality. The taste will be different, the pickles taste will be different, the spices taste will be different. So this leads to variability of the taste in the quality of the material, right? So the consistent incoming raw materials is not there, right? Inconsistent measuring vessels is also not there at home, because we generally see speak 1 teaspoon, 2 teaspoon, but my teaspoon of home may be different from the other home teaspoon, or whenever I am cooking first, the teaspoon which I am using, it maybe in the another time I am not using the same teaspoon. So this measuring vessels are not consistent. So but in industries we have to be consistent, like the in raw materials which are incoming, the measuring vessel which we are using, it has to be defined, specifically defined, otherwise we will not be able to maintain the quality. So this, from this particular examples, I want to say, like, what are the things which we need to control for the consistent quality, so that we can consistently get the same quality. So this is by the food I am saying, because in food it is very difficult, but in pharmaceuticals we have to, if we require the consistent quality, we have to work upon this particular things. We cannot use the raw materials from different vendors and developing the technology. The raw materials has to be from the consistent supplier or a reputed supplier, right, which can supply, and the supplier should also be registered into the regulatory country. That is a requirement very much. Inconsistent process, like soaking time is one of the example. Like we generally soak for 20 minutes first time, but next time we are doing for 30 minutes. So processes or methods are not defined properly at home. Like there's YouTube videos generally, like when we do food, we do not have the so much control on the quality, like soaking every time 20 minutes only. No, it is not like that. So we generally soak for, when it is mentioned 20 minutes, we generally soak for 30 minutes, for 40 minutes also, like. So in inter-manufacture variability is also like the type of cutters, type of burners, flame intensity, utilities variability is also there, room temperature, room humidity, water quality. Like if you are taking water from your RO system, aquaguard systems, or you are taking the directly from tap, the water quality is different, and you are getting the different taste in food. So this is, water is also one of the incoming raw material into the formulation, into your food component. That is because of the difference, we are not able to get it consistent food. So these all things has to be controlled very much into the formulation, pharmaceutical formulation. So this is very good example, like what gives us the insight that what needs to be controlled within the technology transfer. So for the pharmaceutical research product, the two things which we need to prove to the receiver unit by the sending unit is that the formulation which they have developed, or the method which they have developed, the process they have developed, it is nothing like, because basically I'm going to speak more into the formulation part today, but it is the process transfer also, the method transfer also, which is we can do from academic to the industry. Uh, so the product which we have to develop, the sending unit has to prove that the product is safe, the product is efficacious, right? And with that, they have to build the quality of the product. The speed is also very much important. The result, the testing is very much important, and the one very important parameters there should be the cost reduction, because if the companies are extensively working on the cost, they are working on the cost reduction. That is one of the projects, one they are doing it, that they are reducing the cost of incoming materials, utilities, like they're working extensively on that. They want to work on the processes where there is a less of processes, like uh, you can take an example from
cost perspective, like with the wet granulation, where you are granulating using the water or solvents, and then you are drying it. So there is a drying principle. That there's one drying principle in between. The sifting is there, the blending. So these all are there in the wet granulation. But when we go to the direct compression method where we are using the excipients which are already granular in nature, we need not to make the granules. We are just adding the API inside that and then just only do the compression part after the blending. So this only two steps process, whereas in the wet granulation process, it is a 5 to 6 step process. So this number of steps decreases, and the complete in a day, if we can take 4 cycles or 3 cycles, 3 production cycles, with that direct compression method, we can have 7 to 8 cycles in a day. So that gives a almost a double of output in a day. So this is the very major importance of cost. So we have to think that particular from that particular respect also from the cost angle. So what documents are required for to prove that the product is safe and efficacious? Because these are the documents which I'm going to speak. It is very much required from the sending unit. What are the mandatory documents we are going to see? Like how we have select the selection of excipient level. How we have did the selection of the excipients and its level. It is very much required to prove the safety of the product. Impurity level, we have to prove that what is the impurity level into the finished product, and during the stability also. So minimum 3 months stability generally we need to give to the industry before giving the technology transfer, because this gives the maximum confidence. This stability data is one of the data where I can see, like, most of the colleges, they know the technology, know how they make some formulations, but they never generally, like, some people lack, they do not do the stability studies, and they want to propose that particular technology to the industry, but industry generally do not take that particular technology without the stability data. So this is one of the very important data which is required to give the confidence to the receiving unit, that is to the recipient of the technology. One very important thing, if it is a liquid, tablets it is generally not, but if it is liquids, if it is semi-solids, if it is then microbiological enumeration test is very much required. Even in the tablet solids also it is required, but why I'm saying in tablets it is not very much critical, because we can control with the incoming materials. But now in the microbiological enumeration test, it is very much required, because in the liquid dosage forms, the semi-solid liquid dosage forms, the chances of microbial growth is very high. So they want the confidence, the receiver unit has want the confidence on this. That is why this particular data is very much required. If we are using liquids and all, and if we are using preservatives to control the microbial growth, then definitely preservative efficacy test is also required. It is one of the optimization techniques to optimize the preservative level, because low level of preservatives generally after some time, maybe after 1 month or 2 months, the microbial growth increases. So this preservative efficacy test is very much important during the in use of formulation. To prove the safety of the product, residual solvent test is also required. When if we are using the organic solvents into a formulation, that has to be removed from the formulation, or the minimum use of that residual solvent should be there, because it is can be carcinogenic, can be toxic to our body. So this test is very much required, but it is not mandatory, because this particular two things can be optimized. Toxicity data, it can be done by the receiver unit. It is not required by the sender unit to do all this test and then produce. So the very mandatory requirement, which I can say from all this, is the 5 basic things which we discussed, out of which the stability data is very much required. Like if you are doing for the tablets also, minimally generate the impurity level, the hardness of the tablet, and the dissolution profile at each and every time point. And if you have minimum 3 months and you have the confidence, then it will be helpful for the technology transfer. To prove the efficacy, one is the dissolution data, that is we have to be similar. The F2 value should be similar with the reference product. This is very much required. And if it is transdermal product, the flux data is very much important. If it is topical, the skin permeation data is required. Because in there, the drug release data and the dissolution data is not very much important. The flux data, because it has to penetrate through the skin layers inside the blood circulation in the case of transdermal product. So this flux data is very much important. If you have some in vitro efficacy tests like in situ gel formation, there are certain eye ointments which are liquid in nature, and then when we inject into the eyes, it is generally converted into the gel. So this, when we apply it into the eyes, it is converted to the gel, and this data is very much required by the in vitro testing methods. Clinical studies is also required, like bioequivalence studies and all, which are required, but they can be done by the receiver unit. So that data is not mandatory at all to do by the sending unit. So the technology transfer, the team which is there between the pharmaceutical development and the manufacturing, the technology transfer is there, where we are basically involved into the scale up of the formulations and taking the TPQ batches of that, the process validation batches. If these process validation batches are satisfactory, we say it is almost, the technology is completed. Now it is a continuous improvement after the into the manufacturing unit, by the R&D unit, if there is certain problems which are arising uh, on the day-to-day basis. Whenever we are developing a product, whenever we are doing some formulation development, then certainly QTPP and CQA to be defined first. What is our requirement, that has to be tabulated, and this particular data is also required by the receiver unit during the technology transfer documents. What are the targets which are there? So these are all targets. The QTPP we are going to list, like this is the one of the examples for the tablets, where we have dosage form, potency is there, what strength we are going to develop. It should be pharmacokinetics immediate releasing, Tmax in 2 hours, then appearance, assay, impurities, water content, content uniformity, friability, dissolution, disintegration, microbiological. But all these target QTPP are not very much critical for the formulation. There are certain aspects which are very much critical, which we have to consider, like the water level, which is not very much critical if the API is not sensitive to hydrolysis. If the API is not critical, then no need to like have a very much control on the water level, like very strict limits are not required. Then you can have not more than 1%, 2%, 3% into the formulation, right? If the API is very much stable. So this water level becomes critical if the API is sensitive. So here it is, the non-critical part is the dosage unit and the potency, which is almost, we know that it is not very much critical because it is target. We are going to make it. It is not very much applicable, very much critical to control that particular parameter. Like hardness of the tablet is very much not important, because the hardness is directly have the impact on disintegration. So and dissolution. But if we are doing the dissolution and if we are proving it, it is within the specifications which are designed. So this dissolution is very much critical. So if dissolution is critical, then disintegration and hardness does not play a very much critical role in design of the formulations. Now we are going to discuss factors which are going to affect the QTPP of the formulation. So there is 6M. From the first M is methods, we can say. Whenever we are doing some process development, we are doing the method development, it should be scalable. Means what? Whatever we are doing into the lab, we should think of that particular process should be having the same scalability. It should be having the same principle of working into the industry. It should be scalable up to the 1 million tablets or 5 million tablets. So this is somewhere we have to think of that particular aspect. How to do the technology transfer. Then development as per the RU machine suitability. If you know that, if you have got the project, like I can say one of the examples, like the first during the introduction, Mr. Junaid Tantre has told that I did the first development of the first indigenous transdermal patch. That was my PhD project, and that was developed in the lab itself. And there was no Indian company which are manufacturing the transdermal product in-house. They do not have the facility of production also. So was the first project which we developed into the laboratory, and then we scale up into the industry. Even the scale up machines are purchased by our team, my team. I was involved in that. So we know how to, and this is possible only because in the itself in our laboratory we have the machines to produce the transdermal patches. It is only possible you so that is why I mean to say the receiving unit and the sending unit should have the machines of the same principle based, so that the technology it will be easy for the transfer. A design space to be properly defined for the process on the process performance, like up to what process parameters, what is the drying temperature we need to keep. Like if you are drying it at 70 degrees Celsius, it is safe. But if you are going to dry at 80 degrees Celsius, the drying efficiency becomes improved. The drying time also decreased, but the impurity level increases. So that is why at 80 degrees Celsius, it is not uh, good enough for the drying of our formulation. So that design space we have to make, under which space, what range this particular formulation can be processed to get a stable product. Very good example, like we should have the end values of any process. Whenever we are making any process, we are developing any process, any method, we should know when this process is ended, when this process is completed. Like uh, we should know that. So how we have to control our process, we should have the control on our processes. Like if you are doing a drying process, how we are going to say whether drying is completed or not? Basically by measuring the LOD, loss on drying, on the granules. If the loss on, like if we already said like the loss on drying should be between not more than 3%. So when the LOD value comes below 3%, that gives us the assurance that the drying is completed. So that means whenever we are doing some process development, we are developing this process, we should give some end values to our process. Like if we are doing homogenization, if there's some coarse material into the semi-solid or in the lotion formulation or in the liquid dosage forms, we are doing some homogenization step. What we need to understand, why we are doing the homogenization, to understand the last endpoint is the particle size distribution. We should know what is the particle size distribution we should get. So I'm going to speak more on the particle size distribution in one of the slides, which is going to come next, to give you the more proper insight on that. If particle size distribution is difficult to be measured because certain colleges or certain laboratories does not have the apparatus to measure the particle size distribution for the liquid dosage forms, then I can suggest that you at least measure the viscosity of your formulation. What is the end point of the viscosity? Because viscosity is also dependent on the particle size. If the particle size of the excipients, API result into the have the great impact on the viscosity. So you should have the control on the viscosity or either on the sedimentation rate, because sedimentation rate also gives us the good picture on particle size distribution. So if you do not have one thing, I want to say here, like you can think alternatively how you can control the process. So this is by this particular statement, I want to highlight that processes should be precise. Precise means it should be reproducible. Every time we should get the same sort of results. So what do you mean precise? How we are going to measure the precision? It is only by using a mathematical tool which is known as RSD, relative standard deviation. If the RSD is low, that shows that the precision is high, but you are getting the drying process in a similar fashion again and again, right? So one of the very good examples which are mentioned below, if you are drying at 60 degrees Celsius for 30 minutes, your mean LOD is 3. Your limit was not more than 5. That is, it is LOD is almost reached. It is 3, but the RSD is 3.2. But if you are drying at a high temperature, 70 degrees Celsius, and only for 10 minutes, you are also achieving the LOD below that. That is 2.8. But the RSD is very high, 5.8. So out of two processes, if anybody wants to say me which process is better, so I will say the drying at 60 degrees Celsius at 30 minutes is much more better because it is giving me the consistent results. Whereas 70 degrees Celsius for 10 minutes, the mean LOD is less, but it is giving me the non-consistent results. The RSD is very high. So precision should be there in your process. I hope this is clear to you. Yeah. Methods, when we are going to discuss the methods again, to get the more uniform blend, if we are blend uniformity is an issue because when your API quantity percentage is 1 or 2% or less than 10% in the formulation and 90% is your excipients, then it is very difficult to get the blend uniformity for the API. So generally during that time we use the geometrical or sandwich mixing methods. Cleaning method is also required to be developed by the sending unit because how to clean the vessel after the manufacturing, because otherwise it will give the contamination to the next batch which is of the same API. So this cross contamination should be avoided by cleaning method. Cleaning method has to be developed by the sending unit and it has to be validated before the transfer to the receiving unit. Analytical method should also be validated because whatever methods you are using, it should be validated before taking the exhibit batches. So like it can be validated at the receiving unit itself also. It is not required that scale up sending unit should validate the methods. Receiving unit can validate the method. You can take the scale up batches before take exhibit batches before taking the exhibit, or you can say it is exhibit batches can be process validation batches also. So before that we can validate on the scale up batches before on the pre-exhibit batches, the analytical. So it can be done on the, I generally prefer it should be done by the receiving unit. One of the important example which I want to highlight on the complexation method, where I have extensively worked during my industrial uh, tenure to work on the cyclodextrin and the ion exchange resins complexation formulation for the taste masking and to improve the dissolution. The main method parameters to be optimized, we should know the method, what are the main critical method parameters which need to be optimized is the optimization of the time, because the soaking of cyclodextrin, the mixing of cyclodextrin with the API is very much important. The volume of the diluent, because it varies the percentage of API and cyclodextrin to the formulation. So this the diluent is very much important. The stirring speed is very much important. If it is ball mixing, the ball mixer speed is very much important. The pH, if we are doing the complexation by the slurry method, the pH is very much important, specifically in the ion exchange resin complex formulation. So this process parameters, the method parameters has to be optimized, and we should give a certain time that it has to be done in certain 30 minutes, not less than 30 minutes. We should give the values to this particular all method parameters before doing the scale up. During the scale up, generally these values get on changing because working in lab and working in industry, generally these parameters has to be re-optimized onto the larger scale. But we should know what parameters has to be optimized again at the larger scale. This is one a very good example of cyclodextrin complexation, where a team has made the amorphous ternary cyclodextrin nanocomposite of telmisartan for oral drug delivery. The purpose, the problem statement was to improve the solubility and reduce the pharmacokinetic variability of telmisartan. If you see telmisartan in the fasted marketed product, it is blue. In the fasting condition, it is a very good bioavailability. The plasma concentration is very high. Whereas in the fed condition, the telmisartan bioabsorbance is the plasma concentration is very, the bioavailability is less. So because of the pharmacokinetic variability during the fasting and the fed condition, the telmisartan was generally then it was advised to take in the fasted condition before the food, right? But there is a lot of impact on the fasting and in the fed condition of the marketed formulation. So that variability, the company approached us, and then we make a cyclodextrin nanocomposite for that, and the results are here. The team has made the telmisartan ternary complex using beta cyclodextrin and meglumine, and we did a high pressure homogenization of formulation, and then due to high pressure homogenization and the ternary complex, the crystalline this is the first figure you can see below. It is a API telmisartan is crystalline in nature. It is converted into the amorphous form, that is C figure, which we can see amorphous nanocomposites which are formed. So this is a very good example. And the B figure, the central figure, it is only with the cyclodextrin. But when we do the ternary, it converts more into the amorphous form. So now we can see the uniform pharmacokinetic performance in the fast and the fed condition, where both the now the marketed product is behaving similar. So this is one very good example of technology transfer from my team to the industry, like where we worked upon the amorphous nanocomposite and did it by high pressure homogenization. Now I come to this is for the formulation angle. Now I'm going to speak more on analytical methods also, because some of the professors or some of the people will be much more interested, like the analysis. Both the I'm going to satisfy both teams, formulation as well as analytical team also, with respect to dissolution. And there are certain things which I want to clarify by this particular thing, because there is a lot of uh, wrong understanding using the dissolution test method. So whenever I ask about the dissolution to the students, to the forum, okay, why dissolution is developed, so they generally say like to to show that how this particular tablet is going to behave in vivo into the inside the body of the uh, person. So if I'm saying like if your tablet is releasing 100% API, 60% API in 12 hours and 100% API in 24 hours into your body, in vivo, in vitro, sorry, into the dissolution apparatus, is it going to behave the similar in the similar fashion in vivo, like 60% after 12 hours and 100% within 24 hours in your body in vivo also? So they say yes. So the answer is absolutely no here, because in vitro, in vivo correlation is not required in dissolution. If it is there, that it is going to be the best method, but definitely not required at all. So basically, dissolution method is developed. It is a critical quality attribute for routine quality monitoring of batches. It is only the QC test, because whatever specifications you have designed for the dissolution, each and every batch should complies to those specifications. It is nothing to do with your in vivo performance of your formulation. For example, I can say once a day tablet, OD tablet, can have dissolution profile of 2 hours. You do not need to have the profile of 24 hours, because the dissolution profile generally depends upon the media which you have selected. If you have did the media, you can have the same OD tablet profile for 24 hours by using the different media. So it all dissolution profile is dependent on the dissolution media which you are selecting, the dissolution RPM of the stirrer. So by modulating the dissolution profile, you cannot say how it is going to behave in vivo by the in vitro dissolution test. So OD tablets what can have the dissolution profile of 2 hours. The implant of 6 months can have a DR profile, drug release profile of 6 hours. No need to have the 6 months drug release profile. If it is going to release the drug implant in your in vivo system for 6 months, but it can have a DR profile of 6 hours. 6 hours tablet can have the profile of 24 hours in vivo, or 6 hours tablet in vivo can have the profile of 24 hours. Like you have the method of 24 hours, but in the body it is releasing all the 100% in 6 hours. So all these are possible. So these are only a quality control test for the uh, monitoring of the batches. It is nothing to do your in vitro method with the in vivo method. This is very important to understand, because this is something, a lot of times when we discussed with the people, we got the wrong answers. So to clarify to the team, to the uh, people who are here as an audience, I think participants, that this is very important point you should consider, that uh, then what is much more important for the dissolution method? If it is not behaving, if the correlation is not very much important, the very important thing is it should be discriminatory in nature, the last point. But one more important point which is there, it is used as for the exemption of biowaiver. It is used as an maybe used as a exemption for the in vivo bioequivalent studies, for basically for BCS class 1 and 3 molecules which are highly soluble. So we can have the biowaiver. We need not to do the in vivo bioequivalent studies if we can prove through dissolution data. So this is also one of the important tool, the dissolution test, through which we can get the biowaiver. Because if we get the in vivo bioequivalent studies, it is more than a crore of rupees to involve in the in vivo bioequivalent studies. So we can save that amount if it is the molecule belongs to BCS class 1 and 3, which I'm going to speak in the later slides also. And dissolution profile comparisons, it should be discriminatory. uh, what do you mean by discriminatory? Like, here we need to see F2 value, that is a similarity factor. The similarity factor should be between 50 to 100 if it is similar formulations. But if we are designing one bad formulation where we have not used the excipient, if we have used this very high level of polymer, and the good formulation where is the optimum level of polymer, they should be discriminate by using this. So if they are discriminating means F2 value should be below 50. F2 value should be below 50. If that means they are discriminating, the good formulation, the bad formulation is discriminating. And if the value F2 value is between the similarity factor, that is between 50 to 100, that means both the formulations are similar. Whereas my one of the formulation is bad and one of the formulation is good, but still they have the F2 value between 50 to 100. That means that dissolution method is not good, because it is not able to discriminate between the two formulations. But if the dissolution method able to discriminate, that means F2 value is less than 50, that method is much more acceptable by the regulatory agencies and easily acceptable. So this is about the biowaiver. The inclusions is by BCS class 1 and 3 molecules. Is we can prove through dissolution test. Different salts are also acceptable. Like one, if the different hydrochloride salts, hydrobromide salts. So this salts, if it is one the same API is having the different salt forms, that is also we can include into the bio waiver. Prodrugs can also be considered. Only the things which are not considered for the bio waiver is the BCS class 2 and 4 molecules, different esters of the same API, different ether isomers forms, and buccal or sublingual absorption formulations are not considered into the bio waiver usage form. How to design the dissolution points, time points. So this is one of the very good example. Like if it is BCS class 1 molecule or BCS class 3 molecules, we can give a single test point not less than 85% Q in 15 minutes. Or if the Q is less than 85, we can have multiple time points also. But for specifically for BCS class and 2 and 4, we have to give the multiple time points. Multiple time points mean 3 or 4 time points, like how much is release 40%, how much 60%, how much 85%. So this multiple time points we can give. There we can have up to 60 minutes, 90 minutes. No need to give, because of BCS class 1 or 3, we expect that they should be soluble Q 85% in 15 minutes. But in BCS class 2 or 4, where they are less soluble, but they are conventional immediate release, you can have the dissolution profile of 90 to 120 minutes also. For the extended drug release dosage form, that is controlled drug release dosage form, the first time point should have the release of 20 to 30% or around 1 to 2 hours to prove that it to assure that it is not a premature drug release is there from the formulation. Premature drug release means like dose jumping, we want to say. It is not dose jumping from the formulation tablet. The complete dose is released because it is a controlled drug delivery, so it has to be control in a controlled fashion. The second time point which we can give, which is around 50%, and the third time point which is at least 80%, more than 80%. So that third point which we can give as a, so minimum 3 time points is required for the extended release dosage form. These are the regulatory requirements. The second is a material which can contribute to the quality of the product. So the critical material attributes we have to understand, which can influence the process of the product. Like one of the examples is diluent mannitol. Mannitol is not very much critical if you are making the conventional release dosage forms, but it becomes critical if you are designing the oral disintegrating and mouth dissolving tablets. So it has a very important role, the mannitol. The particle size of that mannitol, particle size distribution plays a very important role of mannitol if we are preparing the oral disintegrating and mouth dissolving tablets. Similar, the second example which I have kept here, it is the BET test, which is not very much important for the excipients used for the non-sterile dosage forms. But for the sterile dosage forms, all the excipient, the BET test should be preferable because that gives us that the pyrogen, it is the formulation is going to have the less BET contamination. That is bacterial endotoxin test, BET. The first very good the example which is highlighted is here is the one case study, that is the molecule API is belonging to BCS class 2, that is a less soluble molecule, and it is more and it is sensitive to light and oxidation. The question is, what will be the CMA for API? What is the critical material attribute for the API? Because API belongs to BCS class 2 molecule, it is less soluble, so which should be the controlling parameter which gives us the consistent performance of that particular API into the products. So the answer is particle size distribution. The particle size distribution has to be uniform. So how we measure the particle size distribution and define the particle size distribution is by the D value. So D value generally gives us about the area of the particle size distribution. So generally the D value is mentioned as D10, D50, and D90. D10 means the 10% of the particles are below that particular particle size. Like there are two examples, A and B. There's powder API A and the API B. The D10 is 1 micron, means 10% of the particles are below 1 micron. Here B, that 10% of the particles are below 10 micron. D50 means 50% of the particles are below 12 micron, and here the 50% of the particles are below 30 micron. In the third one is the D90, where 45%, 90% of the molecules, the drug particles, are below 45 micron, and here it is 90% of the particles are below 50 micron. Correct? So D90 means 90% are below 45 and 50 micron respectively. If you see both the things, both the A and B, we can have the inference like this. A is finer because if you see 90% are less than 45, and here 90% are less than 50. So A is finer. If you see D10 and D50 value also, this is less than molecule B. But if you see B is more evenly distributed, the values, the difference between the three is between 10 to 50. The only difference is 40 micron. But here the difference is from 1 micron to 45 micron. It is a difference of 44 micron. So differences is more. So B is more evenly distributed, whereas A is almost finer. I hope this is clear to all. So how it reflects into the API, like this is one of the example of COA from clonidine USP. How they give the values in COA, like you can say D10, D50, D90, they have mentioned the specification limits is not more than 15, 30, and between 30 to 90. Means D90 is should be between 30 micron to 90 micron. Another question is, what is the CM? Now we have discussed about the CM of API, but what is the CM of excipients, which is very important. So that is the second point. That is, it is sensitive to light and oxidation. The molecule is sensitive to light and oxidation. So what can be the CME? So light is from the environment, but oxidation can be done by through the excipients also. So we have to control an excipients which can result in oxidation of my API. So it is, we should select an excipients which is having the low peroxide grade type of excipients which you should select, or otherwise we should add antioxidants into our formulation. So how to select an excipients? What excipients to be selected? What percentage of excipients to be selected? That is through IID limit. Generally by US FDA, there is an active ingredient search database where we can have the API, every excipients used for which dosage form and their maximum potency per dosage unit dosage is already mentioned. So always use in a formulation, always refer IRD database for the selection of quantity of your excipient into the formulation, because that is much more acceptable, because it is already proved that concentration is not toxic to the body. So this is, if we refer, if our concentration is below this level, like it is, if I am making a coated pellets which is going to be used for the capsule, and I am using the copovidone K2831, and it is below 11 milligram, so my formulation will be easily accepted by the receiver unit. So we also should always refer the IID limits. And one more important thing which is there, if it is not there listed into the IID, then the second thing which I just want to say, like use the handbook of pharmaceutical excipients, which also indicates what is the percentage which is generally used by the formulators across the world into the formulation which is not toxic. So the handbook of pharmaceutical excipients also gives the values, gives the numbers which is toxic in nature. Like if I am going to see what is low peroxide grades, like there is an example. The first is the Povidone 30, which is normal grade of Povidone K30. There the peroxide limit is maximum 400 milligram per kg. But if you say Colidone 30 LP, the next COA, that is a low peroxide grade, there the peroxide level is very much controlled. It is the maximum unit is from 400 to 150. That is, if they are controlling the peroxide, it should not be below, it should not be above 150 milligram per kg. So if your API is very much sensitive for oxidation, you can use the low peroxide grades excipients also, which can control the impurities into the formulation. Now, the third example, what to be precautions for the manufacturing? You can use the monochromatic light into during the formulation. And if it is oxidation from the environment, if it is liquid dosage form, if it is injectable dosage forms, generally in parental dosage forms, we do the nitrogen purging also. Nitrogen purging, due to the nitrogen purging, how much nitrogen purging to be done? So just like as I told you that every process has to be controlled. It should have some end value. So then how much nitrogen purging we need to control for 10 minutes, 5 minutes, what is the pressure of nitrogen, all these things, how we are going to control. The end value we are going to get is the DO, that is the dissolved oxygen. So if you see, the dissolved oxygen meters are available into the market. One is tabletop models, one are portables one. You can have different one from this vary from 5,000 to 1 lakh rupees depending upon the company's models. And you can have the DO limit. So you can define what is the DO, which is not more than that. It should not be there into your liquid dosage form or parental dosage form. So you can control, like you are controlling the pH of your formulation, you can control the DO, dissolved oxygen of your form into the liquid dosage form. So that is basically DO, it is for the liquid dosage forms, and specifically for sterile liquid dosage forms, right? The third one is the measurement. generally measurements, we whatever we are doing the method, it has to be measured, because the CQAs we have to measure, right? So use a measurement technique which is more specific and sensitive. Which is more sensitive and more specific, I'm going to give you an example here. You can do the analysis by UV spectrophotometer. You like, this is the extraction principle. Lot of extractions are going on here, and then it is filter through the filter, it is going inside through the circulation of peristaltic pump into the different spectrophotometers, right? So which one is good, which one is good, going to give us the most sensitivity and specificity? So when we are going to say it is UV is very much less specific, I can say, because when we are doing through the UV cuvette, it is completely doing the analysis. It is not very much specific, because in cuvette you have the bulk solution. So if there are two extractables from this particular extractor unit, so they can both interfere. But if you have NIR, if you have HPLC, they are more specific. Now during the during the process analytical tool, which is there, it is NIR analysis, which is most of the companies are working upon that now, where you can in during the process itself, you can measure from 5 to 6 different locations, whether on the basis of absorbance in the NIR, how is it uniformly mixed or not. So this is NIR analysis is also there. But I am saying this is one of the tool, that is not the process. This is nothing relevant to the technology transfer process. In your lab, you can do the analysis by HPLC, by UV, right? If it is only one API, or if it is the method is very much specific, right? Here I want to say one of very good example of transdermal patch. Like the transdermal patch, the skin permeation was done. This is, I can say, this is during my first initial year of 2003, where I started working on transdermal patches, and this is the practical example. And uh, the skin permeation was done using the France diffusion cell. I used the skin, rat abdominal skin, and then I did the permeation studies of my developed patch and the reference product. And in the receiver compartment, whatever analysis we did in the media which is there into the receiver chamber by the UV, and we get the high absorbance in both in reference and test product. So this is, it's indicating that it's a very high absorbance, which is not expected also. In that much amount of drug I have not added also, but it is giving the very high absorbance in both reference and test product. So basically, this is why, because skin proteins leach into the solution, into the receiver compartment, and they also contribute to the absorbance. So I was analyzing it through the UV. So UV is a non-specific method, because it's in the cuvette you have the complete solution where it is all the proteins also interfering. So should not be used for the skin permeation studies. Then we have to use a very much specific method, which is HPLC method, where it is a chromatographic method where skin proteins and your API get separated from each other, and then we do the through the UV analyzer. So I always use some HPLC method. That is, I am going to say the measurement should be specific and sensitive. Here, the point which I told you, like I'm going to tell you with one of the examples, like here, UV method should be avoided. It should not be used for skin permeation studies. Important CQAs for TDS, which we need to measure, TDDS, that is Transdermal Drug Delivery System. The very important thing which is the uniformity of content and assay, which is there into the tablets itself. But very unique thing is permission enhancer content, because in the design of transdermal patches, we are incorporating permission enhancers also. So certain times we need to analyze the permission enhancer content up to the shelf life, up to the, that is 24 months, 36 months. the permission enhancer content also we need to analyze, because if the permission enhancer content decreases, so if the user is using my transdermal patch, the designed transdermal patch, it can give the less permission, because permission enhancers also degrade during the stability. So it is always said that permission enhancers content should be controlled up to that specified limits, within the specified limits. Drug release, in tablets we are generally using USP 1 and 2, but for the drug release transdermal patches it is USP 5, 6 and 7 which are there. And because these patches are going to stick on the skin, so we have to do some physical addition test also, so that it will remain on the skin up to during the period of usage. So there are 4 parameters which is there: peer addition, shear, tech test, and release force. These 4 tests has to be done using a universal tensile tester. This figure shows that it is universal tensile tester to be used, which is used in industries, and they are into some colleges also available. Impurities, uh, and if you like, there is no, certainly all the facilities should not be available at one point, but like if you say ICT, NIPERS, IITs, they have this type of operators, so you can collaborate, or you can on the paid basis also you can get your testing done. So there are certain analytical laboratories where you can pay and then you can did it. You can do your testing. Residual solvent content, because the transdermal patch contains generally manufactured in or using the organic solvents. That is why we need to control the residual solvent content with the finished formulation. The microbiology enumeration is also very much important. If you are using some water or the water is there, so that is total aerobic microbial count, total yeast mold count, S. aureus, Staphylococcus aureus, Pseudomonas aeruginosa. These are the 2 microorganisms should be absent into your formulation. But if you see the tablet dosage form in oral preparations, these 2 things are Pseudomonas aeruginosa and Staphylococcus aureus is not mentioned, because orally E. coli has to be controlled. It should be absent. It should be in oral formulation, like whatever tablets. Then these 2, because these 2 organisms basically attack on the skin and then do the skin infections. But they are, when you are taking it orally, if these 2 or microorganisms are there, they are not going to hamper or cause any disease to your body. But E. coli, if it is there, it causes dysentery, diarrhea, etc. So what are the parameters which to be controlled? That is why the sending unit should always have the idea what things they have to control into the final formulation, into designed formulation. So here's the difference between the transdermal patch and the oral preparations. Now, the very recent concerns which is arised from basically from 2020 onwards is the nitrosamine impurity, which has to be controlled to the formulation, because it is one of the probable human carcinogen. So to control this formulation, FDA said that manufacturers are responsible for understanding their processes and to control their limits. So FDA has published the guidance just recently in August 2023 on the intake limits for nitrosamine drug substances related impurity, which is also known as NDSRIs. And if you can see, there are 2 examples in 2020. There's a lot of companies have voluntarily recalled the metformin formulation, because metformin formulations and ranitidine formulations, these are the 2 important, and many more also, where the nitrosamine impurities was found above the level which can be carcinogenic, which can cause cancers. So that is why there is now very strict control on nitrosamine impurity into the finished formulation. So that has to be controlled through the raw material which you are using. That has to be controlled through the water. Water is also one of the important uh, raw material which can contribute to the nitrosamine impurity. The water quality is also very much critical here to control for the nitrosamine impurity. Now we come to the man. If you are coming from the man, then manpower training from receiver unit has to be trained from sending unit, because sending unit they are more technical people, because sending unit is designed the formulation. They did the innovation. They know the in and out of the formulation. So this can be done through PowerPoint presentations, process demonstration at sending unit. In this itself into the laboratory, you can demonstrate the process by the receiver unit. Transfer protocols are designed. Then we need to have the quality and qualified experienced manpower in the receiver unit, because if the manpower is not technical itself, they do not have the uh, ap- appropriate degree, like they are not b.pharms, m.pharms, then they are not able to, it will be difficult for them that to understand the, that is why it is known as qualified and experienced manpower is required into the industries to receive the technology also. Now coming to the Milayu, which is the fifth M, which is known as the environment. So environment is very much important to get a very consistent quality product, because all these are related to QTPP, how to control the QTPP. So now if we say the differential pressure requirement, if you say if it is pressure zoning is very much important, because if you are designing an oral dosage form and making manufacturing the oral dosage form, the pressure differential should be negative with respect to corridor. The area, manufacturing area, should be negative with respect to corridor, because the powder should not be go inside the corridor. Otherwise it will contaminate the corridor. So the powder should, the API should be there inside. So we should know the requirements. Nitrogen, as I told you, that DO, nitrogen controls the DO. So what is, how much pressure is required of nitrogen, and what is the percentage purity of nitrogen is required. This all is very much important in the with respect to utilities. Humidity is very much important. If your tablet formulation is sensitive for the with the, is very much hygroscopic, it absorbs the moisture and then change the color of the formulation. The humidity is control is very much important. And if you are making oral disintegrating tablets, then humidity control, humidity generally in industry it is below 60%, but for oral disintegrating tablets, for oral films, that humidity conditions is more stringent. Generally, it is not more than 40%. So there is a difference of delta of 20%, like not more than 60% in case of normal tablets, and here it is not. Area qualification, you are like class D, class B, which area should be qualified for the production. So all these are playing very much important role for the sender unit. Like, but you should know what is the humidity and all. It has to be controlled. You should, in the planning, you should have, if this much temperature and humidity, my product is stable. This product is stable. So this is gives the idea for the receiver unit that I can control this humidity up to 40%. Otherwise, if they are not able to, then they have to install the dehumidifiers into their facility. So all those things has to go through the sending unit to the receiver unit. This particular points on the environmental requirements. The last M is machines, which can be very much responsible for the quality of the product. Sending unit and the receiving unit preferably have the same principle of working. I am saying it is the same principle of working, preferably, right? Not mandatorily. If they have the same principle of working in machines having the same principle, it is always good. Batch size selection is dependent on commercial requirements, machine capacity. Then this is very much important. You should have the batch size not big, because if you are doing the scale up, because if you are doing the transfer, so don't select the batch size which is too very high, because then the blend uniformity will be difficult. So you should generally do it 60 to 80%. Do not work on 100% capacity of the equipment, because it will be difficult to get the uniform blend, and if it is there, it will take a much more time. So always work on 60 to 80% of the capacity of the equipment, which is good for technology transfer. Cleaning method should also be developed and validated for the machine cleanings. Machine cleaning method should be also developed. One dirty equipment whole time studies to be performed as a challenge studies, which is done at the receiver unit, because we make it dirty and then we try to clean it by using our cleaning method. If it is cleaned properly, that means our method is properly validated. Otherwise, we have to optimize the amount of cleaning agents, cleaning time. This is all needs to be re-optimized. if you say from the lab to tablet, one very good example of tableting machines, which I want to highlight here, like dispensing is like we are doing in the lab through weighing balance, and in the industries also it is RLAF, but weighing balances are, so they are almost equivalent. No change, no gap is there. Sifting, we also are having in the lab the sifters. In the industries also they have the big sifters, they are almost equivalent. The blending process, in the industry we are doing, in the lab we have small blenders, or we are generally doing through manually, like in poly bag we generally doing through hand. But in machine, in industries they have, in plant they have the big blenders, so they are almost equivalent. You can do it, it is not going to challenge much technology transfer. But granulation is very important, critical, because if you are doing through hand, but industries they have RNG, so you do not know the process time, how to control the process, how much amount of, because manually the amount of liquid and the amount of liquid used in the granulator, the hardness of the granules all gets very much changed. So that is why I always recommend that the small RNG in lab should be present. If you are doing the technology transfer, you should have the small RNG in the lab. It should not be, manual process cannot be equivalent. Like here I have said manual blending can be equivalent to the blending through the equipment. Sifting can be equivalent, weighing can be equivalent, but here granulation cannot be equivalent, because it is going to play a very important role during the technology transfer. So that is why I recommend that if any college, any professor, or any institution is working on granulation, it should not do through manual to RMG. Drying process also people were using in the previously tray dryers, but in industry now very few small companies only have the tray dryer, otherwise all the companies have replaced tray dryers through FBD. So that is why I'm saying the FBD should be required in the colleges itself also, should be installed if they are doing the technology transfer, so that to understand the process control and to design the space. Compression machine, always colleges is having the mono compression machine or rotary compression machine, which is more or like equivalent. Doesn't mean if you have the, if you are controlling the hardness of the tablet, then it is more or almost like equivalent. Then we have pan coater, and there is auto coaters, but in pan coater and auto coater there is a lot of difference in process parameters. So I recommend that if you are doing some technology transfer, this is not very mandatory, but I recommend that we should have a small auto coater into the lab itself also. Similarly, the gap analysis for the transdermal machines, the transdermal first is dispensing, second we are making the blending, which is almost equivalent. You are having big blenders, and in lab you are making through overhead stirrer, doesn't make any difference because you are going to make a blend. And in a lab you are doing through petri plate the coating, but here it is a huge gap. The lab coaters, lot of process parameters, lot of variability is there. To understand that, we should have the small coaters, uh, film coaters inside the lab to make the transdermal patch. If we are any professor, any college is required, then they should, doing the technology transfer, they should have the film coaters. Conversion, yeah, definitely you can do through, if you are doing conversion means convert into the unit dosage form, you can do by Caesar, or manual conversion is acceptable at all, is acceptable, because in industry you are doing through conversion through the machine, that is not going to change any profile your quality product, because conversion is only cutting the product into the desired shape and size. So if you are doing through cutting, that is acceptable, you can do in the lab itself, it is not required that you should have some machine. But definitely film coating, because you are going to precisely need to coat the polymer, which is, that is why you cannot do in the petri plate. That is why lab coater machine should be required. So now the overall, I'm going to give the summary of checklist for the technology transfer. That we should have the master formula ready, that is bill of materials, manufacturing work instruction should be ready, cleaning method should be ready, machine qualification should be completed, packaging by the should machine qualifications of what receiver unit should be completed, that we have to check, We need to give the packaging instructions. We need to give them MSDS, if there is any spillage, how to control that, the accident, how to control the accident, right? And the forecast and volumes that has to be given by the receiver unit. Then we should have the list of source and approved suppliers list, specification and certificates of analysis of all the raw materials. Because we are going to make the specification from the certificate of analysis, we have to control the specifications for raw material itself. As I told you, the raw materials are very critical for the controlling the quality of the product. Then we should have the test methods ready, and then we have the primary packaging material specifications in hand. Then these are the specifications like bulk product specification, finished product specifications, stability specifications should be there. We should have the stability data in hand, and we should do the cleaning validation method and packaging validation. That I generally recommend that validation should be done at the receiver unit. The lab should not do it, because this is going to change, and then they have to do the revalidation there. So if you want to do, you can verify into the lab. The 2 or 3 times you can verify, but need not to validate extensively all the parameters and give the validation report. That is not much required. What I am going to say, the art of transfer, basically to the labs, to the colleges, to the professors. This is 1 or 2 slides which I have prepared for them. Develop your expertise in one major field. Like any professor wants to do the technology transfer in transdermal, at least you expertise yourself into transdermal, do many projects, develop your machine, develop your lab into transdermal lab itself. And then don't be do many technologies like I want to do on nanoparticles, nanocomposites, liquid dosage form. I want to say that it will be, because if you see in past also, certain professors have become famous for their own one expertise field. So that is one, that is a suggestion from my side. And if you have developed something, then it's like, if you want to see some facilities which are there, like you can develop a paracetamol pediatric suspension in a college. Like it is a very small project, because, and what you will say ki, what I'm going to do with the paracetamol pediatric suspension. You have made a m.pharm thesis now. Now your student know what is made, how to make a pediatric suspension. But now you compare your formulation with the marketed formulation, and you come to know that your formulation is very good in terms of sedimentation rate and redispersibility. Your formulation is very good, but you are thinking, what should I do? So you certainly you can do. Then what you can do in your periphery, like in like if you are sitting in Delhi, or if you are sitting in, like I am sitting in Ahmedabad, like I can see the industries which are working in Gujarat, and they have the liquid facility, but they do not in their portfolio they have the paracetamol pediatric suspension. So I can offer the samples to them. I can offer that this is a formulation which I have prepared. Don't give the formula, don't give the technique to them, just only give the samples to them, and then it will be easy to evaluate them and show the in comparison, and give them the comparative results of your formulation with respect to industrial formulation, and then it will be easy for them to take your formulation. And very good examples I can say, like metformin glimepiride tablets. People are coming with the smaller tablets. They are using like the controlling release polymer. They are using the high grade viscosity grade so that they can make the smaller tablets. Certain people labs have worked upon improvement of glimepiride dissolution, and they had did the technology transfer to the industries. So this is one of the improvement from the college itself, from the lab itself. So if you know the challenge statement, what are the challenges of glimepiride? It's having the like telmisartan, which I gave an example. Like it is having a lot of variability on the bioavailability. Similar is the glimepiride example. So sustained release formulation you can make. Cosmetics and herbal nutraceuticals I generally prefer, like because it is easily to, like if your lab is making, and then it is easy to give to some industries, and there is a lot of less stringent regulatory requirements is there, and you can easily can commercialize. Then instead of making the API complete API, if this it is basically for the organic scientist people, uh, and the people they are into the synthesis. So they develop the intermediates. From that intermediates, many API can be formulated. So this develop, if you are intermediate, the requirements is very less, and you can do this intermediate technology transfer. You can synthesis of intermediates can you. It is very easy because API control is very difficult, because there's a lot of API controls that requirements is there. That I generally say that develop the intermediates that will be easy from the lab to the industrial level. Stable polymer formation, higher purity. If you know any technique through which you can get the higher purer form of reverse ligament, because it is racemic form. If you can by any technique, you can R form can be separated from the S form, because S form is R form is not very much potent, but S form is very much potent. So if you can have the S form, then it is generally very advisable. So such type of small things in labs you can do, give it to the industries, right? To their R&D presidents, or to the chairman of the company, you can discuss. In the last, I'm going to discuss on the art of transfer from the college to from the lab to the industry. If you have any product development, then generate the scientific data, specifically stability data, prepare some samples, send some samples to the relevant industries which are like you think it is relevant for them their portfolio, and share the scientific data without disclosure of qualitative and quantitative industry. Negotiate on commercial front if they accept your formulation, accept your samples. If they found your samples very good, then negotiate on commercial part of technology transfer, do the MOUs with them, and do the technology transfer. So this is the way I generally worked. And you I also worked with ISF College of Pharmacy, Muga, and did the technology transfer from that particular college. And like as I told you, the first indigenous transdermal patch was the technology was developed by me in the laboratory. That was my PhD project, and we commercialized it. But it is very much possible if you have a good scientific data available. Thank you very much.
Thank you so much, sir. It was nothing short of exceptional, sir. Am I audible, sir?
Yes.
So, yeah, thank you once again, sir. Your expertise and in-depth discussion regarding the aspects of technology transfer. You talk about challenge in scaling up formulations. You also spoke about, sir, regulatory considerations. Also highlighted the industry best practices. So this provide us comprehensive understanding of this crucial process. And thank you for giving real world examples, sir. Vijay. Especially you gave an example of paracetamol, for example, how to start in our own. We can do the comparative studies for paracetamol with the marketed formulation. So thank you once again. I believe that this your expertise, this webinar will be a landmark, and it will be available freely so that many people can go through this. And sir, we have at the end of it, we have received some questions, sir. We have collected the few one. So can we begin the question answer session, sir?
Yeah, sure, sir.
Sir, first we have received that how does the selection of critical quality attributes and quality target product profile impact the technology transfer process?
Definitely, we like the critical quality attributes are the test which is very critical for your controlling the finished formulation, like the final formulation which you are there. Like as I told you, like in the transdermal product, not only the even the assay of API, it is the assay of permission and answer is also very much required. That critical quality attributes means the final testing of the product, which is very much required. Like you have to control this sub-specific microorganisms, which is not required in oral formulations. Oral formulation have different microorganisms which are defined. That is the E. coli. So all those critical quality attributes has to be designed on paper first. What is your like? If you are making some like in a home some, or if you are making something in at home also some dish, you expect that this particular dish should taste like that, right? If you are going for a like if you are going to going into the market to have a pastry, you never expect that it should be salty, it should be sweet, right? So
Absolutely.
on the paper, if you are making any formulation, that it should be, it should you design your QTPP, you define your QTPP and CQA scheme. What is your objective goal? Then only you can do the proper technology transfer. That it is very critical.
Thank you so much, sir. We have next question. What are the major challenges faced during technology transfer? How they can be mitigated?
The challenges which is very much there into because the equipments which are available into the lab and the equipments which are available into the plant, they are very much different in size, proportion. It is almost like 1 lakh time bigger than the lab equipments, right? And their principle of working is can be very much different. So generally, that is why this is the biggest challenge for the formulator, the scientist who is doing it, because that is the biggest challenge for that who do the technology transfer into the industry. Because the
Right.
working of the principles, the equipment is very much different. That is why I say, if you are working into one dosage form, so that you develop your lab in such a way that you have those smaller pilot scale equipments of that particular technology into your lab. So don't work on multiple. That is my suggestion. Don't work on spend on all the equipments. You should be a professor. You should be a technical person for one field. Then only I think it will be easy for do the technology transfer, because then you have the small RNG, then you have the small FBD. Otherwise, people are using the trade dryers and very difficult to doing the technology transfer. Then they generally fail at some stage. So that failures can be avoided if you have the same scale of equipments.
Sir, we have a last question from my side, sir. And as you have worked both in industry and academia, so I think we there are so many conferences, so many webinars, so many seminars going on in the topic between bridging the gap between the industry and academia. I think you are the best person, sir, that can give us more insights how it is to be in the industry and academia, or what are the points that can be noted, and you can specify your journey, sir, shifting from academia to this industry, sir.
Mr. Junaid, this is very interesting question, but the gap is like people are talking about the bridging the gaps, right? Always they are talking about the academics, and but these gaps are made by I can say academics itself, and by the academicians itself, because the requirement is to get the very good papers into the publications, very good publications. Then they generally uh, uh, they like their expectations and the students' expectation is to work on nanoparticles, nanocomposites, and blah blah formulations, microspheres, which is itself a very tough task to complete m.pharm on. In m.pharm practical it is, in m.pharm it is generally 8 months or 1 year is there, or not even over complete 1 year to complete the formulation. So I generally say, if we are want to do like, if you see my technology transfer, that is what Transdermal was a PhD project, but during my M.pharm I did a small tablet technology transfer. I did the technology transfer on oral films, not on my nanoparticles, because in industry, if you see we have to see the market also. In 99.99% of the industries which are working across nearby to us, they want the tablet formulation. They are working on syrup formulation. They are working on. So we should in M.pharm, I want to say we have to bridge the gaps. We have to prepare our students in such a way that they should know the tablet formulation. They should know this syrup formulation. They should know how to control the sedimentation rate, because if it give the because now the mode of teaching is such in a certain manner, they students are working more on the nanoparticles, and later on when they evaluate, they do not have the complete evaluation facility. Also, they are dependent on external agencies, whereas when they move to the industry, nobody is asking from them the nanoparticles, because that company is working on tablets and capsules. So I want to say, like, m.pharm projects basically should be designed as per industry requirements, and the PhD projects should be designed as per the very sophisticated, like transdermal microspheres. There they can work because they have the sufficient time, 2 to 3 years for completion of during the PhD. So that by this way, I think that is the best way of bridging the gap between the academic and the industry.
With this, thank you so much, sir. I hope this question will be this question. It was a question from various others. I hope they all will satisfy with this. And definitely, sir, there should be a change in curriculum also. It's very important that you highlighted this also. With this, sir, today's discussion, we are concluding this 85th SPSR webinar on science of technology transfer from lab to plant. And we extend our heartfelt gratitude to Dr. Gaurav sir for delivering such an insightful and informative session, sir. This will be a landmark for sure. And it's evident that technology transfer is not just about scaling up production, but also about maintaining product quality, process consistency, and regulatory alignment also. Uh, on behalf of SPSR, we sincerely thank Dr. Gaurav Goyal sir for his insightful presentation and to all our participants for their active engagement and thought-provoking questions. We look forward for your continued participation in future SPSR webinars as we explore more advancement in pharmaceutical science. We are deeply thankful to Dr. Shashi Alok sir, National President SPSR, presents Mrs. Monika Sabharwal, ma'am, co-convener for her pivotal role in organizing this event. And we look forward so much once again, sir. It was a pleasure listening to you, sir.
Thank you very much.